Hey, I'm Rémi from the Cell Physics Master in Strasbourg. In this video, I'll talk about fluorescence and explain how it's used as a tool by biologists to study the internal of cells. Cells are composed of many different molecules, and distinguishing them, even with a microscope, is hard, yet it is required to understand how living systems work. Fluorescence is an interaction between light and matter. When a photon meets a molecule, its energy can be absorbed, leading the molecule to an excited state. Over time, the molecule will get rid of this energy and go back to the original state. The most common way is called non-radiative, that is, the energy is dissipated by vibration or rotation of the molecular structure. Sometimes the energy is released by emission of a photon. As the molecule goes from the excited state back to the original state, the energy and the color of the outgoing photon are the same than the incoming photon had. In fluorescence, the emission of photon happens after a vibration. In this case, the outgoing photon will carry less energy and thus have a different color. In some cases, it is possible to block rotations in molecules by chemical means. Let's take this molecule. It is not fluorescent because it rotates easily. Now, if this group has a strong affinity with calcium ion, then when one is present, it will bind here and there, blocking rotation and switching on the fluorescence. Biologists use this molecule as a tool to measure calcium flow in living cells. By injecting it inside cells and monitoring the fluorescence intensity over time, they are able to quantify how many calcium ions enter the cell. Developing more and more environment-sensitive probes is an active field of research and necessary to understand how living systems work.